So I would like to start by thanking the organizers to give me this talk, although I could not make it to Vegas, so I'm very thankful. And I'm going to speak about inferences of cellular networks from bulk, but also spatial transcriptomics data. So as you know, cellular networks play a very important role in tissue homeostasis and development, and also in diseases. For instance, in cancer, the microenvironment, that is all the non-cancer cells that are present in the tumor, they contribute in many ways to tumor progression, but also different resistance mechanisms against therapy. And the major part of the exchanges those cells have to, between each other involves ligand-receptor interactions. So if you are in a single-cell setting, the problem of inferring the ligand-receptor interaction is relatively simple. From the single-cell data, you can obtain cell-type-specific transcriptome, and you can compare those transcriptomes with a reference database containing known ligand-receptor interactions, and based on diverse scoring mechanisms, try to assess what is really present in the tissue and infer interactions between cell types and, of course, specifically which ligand and which receptor. Now, if you are with bulk data, this is more complex because all the cell types contribute together to a single transcriptome or proteome if you work with a protein expression data. But it is also fair to say that although this is more complex and more difficult, there are a number of very rich collections of data sets in the bulk format that come, for instance, with very valuable clinical data that we would like to exploit from this cellular network point of view. And very interestingly, spatial transcriptomics comes in different formats, and in particular, in a so-called multicellular resolution format, for instance, with this very popular platform, Visium, where multiple cells are analyzed together at each spot. That is, in fact, these data are bulk data, localized bulk data. So how does that work? So we propose to evaluate the interaction between a ligand and a receptor by simply considering correlations, but also correlations with pathways downstream the receptor. So basically, a null model of all those correlations is learned by randomizing the data in a kind of a meaningful way, and to report the coherent activity of the pathway with the receptor. We look at the genes targeted by the pathway, plus eventually co-complex. So with this, you can generate tables of putative ligand-receptor interactions related to a certain pathway, which you associate with the usual statistical parameters like Q-values. You can estimate some sort of ROC curves in different data sets. And for instance, what you can show is that assessing the activity of the pathway at a certain depth is definitely beneficial. So you really have a benefit to exploit the pathway information significantly, not to test just the most two correlated target genes. It's better if you exploit more. Now that you have this mechanism, you can associate each ligand-receptor interaction with the genes that report the pathway activity and define a gene signature that allows you to score, kind of determine an integrated score to score the ligand-receptor interaction across tissues and obtain that kind of heat map to compare samples. So if we apply this to two data sets to show how it works, we start first with cancer cell lines. That is, we predict autocrine interactions. There is no microenvironment. These are lung cancer cell lines. You see that those activity scores of the different ligand-receptor interactions, they separate very nicely small cell lung cancer from non-small cell lung cancer, which is expected. These are very different tumors, etc., etc. And the interactions you find are pretty relevant. I mean, some of them, they are pretty relevant to lung cancer biology. But what is interesting is also because we essentially work with those triples, a ligand, a receptor, and a pathway downstream the receptor, you can also reduce the information at the pathway level first, basically like you would do Go-term enrichment or gene set enrichment analysis. And this we exemplify with those transcriptome monitoring the development of different tissues from the embryo to the adult. We compare brain and heart muscle development, and we find brain-specific pathways, of course, muscle-specific pathways, and also shared pathways that are typical pathways involved in tissue development, such as notch, etc., etc. So with this, you have the option to have kind of a summary at this level and then go into the individual interaction at a later stage. There are different visualization tools that are offered by the library. So I exemplify this in this slide on a data set that we published 3 years ago almost, where we found different immune checkpoints. It's a very immunosuppressive tumor, so there are different immune checkpoints. And for instance, you can report the interactions by focusing specifically on a certain pathway, in that particular case, the PD-L1, PD-1. And also, just a few lines of R to show you that this is very, very simple to use. You just need your expression data in a matrix or data frame, and then you call basic functions to prepare the data, and you can immediately infer the interactions, which you can get as a table, but also generate the different plots. You can work with different species because there is an ortholog mapping mechanism embedded in the library. And this is also very simple. Now, switching to spatial data. The thing is, spatial data with this Visium platform, it is basically single-cell data. So the depth is much reduced compared to RNA-seq, and the correlations you observe, they are also on a narrower range. But in fact, it works very well because the model learns by itself the null distributions of the correlations. There is basically nothing to do but to change a few thresholds that are a bit too high for spatial. You have to lower a bit those thresholds, and that's it. So here I exemplify an application of the library to triple negative breast cancer data. So this is the structure of the tumor with different regions, and this is an interaction involved in the extracellular matrix remodeling and this kind of process. This is the global score, integrating everything, the pathway and the ligand receptors. And here you show the read counts of the ligand and the receptor separated. And this is in the invasive cancer tissue another interactions that is related to apoptosis, where again you have the score of the overall structure plus specific information about the ligand and the receptor. There are different visualization functions like this big index that summarizes visually everything you identify. You can also compute associations with the different areas that are color-coded here. And you can kind of project those different patterns because, as you can see, the diversity is more reduced than I would have expected. So we thought that it might be interesting to have this kind of 2D projection to detect basically the main patterns that can occur and annotate them with the interaction. We compare the tool with other packages that address spatial transcriptomic data. That's a bit too long to detail, but basically the message is that we obtain an average number of predictions and the quality is clearly superior. So we define different quality parameters and almost always we obtain significant advantage over the other software. From those Venn diagrams, you also see that the different software, they do not agree too much. So obviously, there is still space for improvement. And in terms of compute time, we are kind of reasonable. Spato is pretty slow. This is Spato. This is STLearn, a bit slower than us, but reasonable. And the fastest is self-funded. Okay. One last example to show reproducibility and coherence in the data. So with our colleague, Andrei Tortoy, we generated spatial data for colorectal liver metastasis, which we analyzed with this tool. And here you see 3 different tumors with very different structures, more or less desmoplastic reactions. So they really are organized differently. And we were interested in the different ligands of EGFR, which we report here according to their abundance. And as you can see, there is quite an overlap between the 3 tumors, although they are very different. There is this guy which is very abundant here that has disappeared, but there is some sort of an explanation, a bit too long for now. Otherwise, this is more or less the same set of ligands. Very interestingly, also, EGFR itself is expressed more or less everywhere in the tissue, but the different ligands, they are more or less specific. So with those region association patterns, you see that some are very specific, like decorin, DCN. You can see it here in the first sample. It's associated with calves, basically. Others, they are basically everywhere as well, and they are in intermediary cases. So you can really map how the EGFR signaling is controlled by different ligands everywhere at specific locations, etc., etc. And the last thing interesting here is that you can use the tool also to discover inhibitory interactions. Decorin inhibits the EGFR signal. And because we had access to these samples and we generated the data, we could validate a few interactions with immunofluorescence. So to conclude, we have developed a library that brings to the bulk format similar functionalities as what is available in the single-cell world for inferring ligand-receptor interactions. It makes those many cohorts of interesting samples amenable to cellular network inference. It is fair to say that when you have equivalent single-cell and bulk data, the bulk data analysis reveals typically 2 or 3 times less ligand-receptor interactions, but those that you only see here, sorry, those that you only see with the bulk, they tend to involve lower expressed molecules, which makes sense because RNA sequencing is much more sensitive than single cell platforms. And it also works very well for multicellular resolution spatial transcriptomics. I hoped that we would have been ready in time, but as usual, it takes more time than expected. So the preprint and the manuscript will be submitted next week. So next week, all that story should be available in details and the code available also from detail. And with this, I would like to thank you for your attention and also all my colleagues and funding bodies who contributed to the development of this talk. Thank you.